that little ring around it with the line connected to it, right? Other part is the central vein, which should be relatively easy to see. None of these are great looking. They're okay, but they're not great looking. See if I can find one that's got a really good central vein. None of those are lovely. I mean, this one's okay. They're never going to be perfect. That one's okay. I don't mind that one. It's got the central little open area there in the middle, right? And then the triad. The triad, of course, we'll look at closer, but the triads are as I explained in lecture, they're in these sections here where you have three of them come together. That's where you have your triads, right? Each one of those has a triad in them. So the next slide should be a close-up of hepatocytes, the cells themselves. So if we look at this one, the smaller one here that we have on the field of view, zoom in on it a little bit. Ugh, that's ugly. These aren't very clear, are they? None of these are really. That's a little bit better. Still not great. Might have to find myself another liver slide. Let's see. Where were the liver slides? All right, well, we'll have to do what we have to do. I'll come back and I'll find a one at some point. Looks like it's mainly reproductive tissue. Okay. All right. Anyway, the good part you can see is you can see the trabeculae of them, right? The little channels of them as they go down, right? The bad part is you can't see the individual cells very well with this stain for some reason. I don't know why they're unclear like that. You can see them a little better there, but they're awfully fuzzy. Tell you the truth, I think they're too thick. So most of the cells you see here are hepatocytes. I mean, that's the vast majority of the cells here. The only other real cell in here is what? Cooper cells, right? Cooper cells are in here. We have special stains for Cooper cells to show them. They're going to be the cells that are not in the plates or the cords. They're kind of off to the sides of it. You can see them without the stain for it, but it is not relatively easy to see on this slide at all. I'll find a better slide for it in a minute. All right, so the two parts we have here, and this is on the test I put up there, right, yesterday. You have the sinusoids and the spaces of Dissay. So the sinusoids are are capillaries, that blood vessels, if you will. The space of Dissay are not. They're gaps. They're just what they say, spaces, right? So the difference between them is you have to look for that lining of the capillary to see that, which one it is, right? And this is about as high as I can go without oil. But even here you can see, if you look, you can see that lining of the capillary bed when you're looking at it, right? For example, if you look right here, you can see that inner lining of the capillary there on that one. Plus, the other key is, what's this thing here? Yeah, when you see the blood cells, that's a real tip-off, right? That you're in a sinusoid, not in a space of Dissay. Spaces of Dissay are usually relatively small, little gaps of areas, like this here is a space of Dissay. It's going to be a little gap of where there's nothing, no blood vessel, no nothing.
What does that do?
Sorry?
Like, why is there a space?
So, that's a good question. It is thought to be areas where it can put out metabolic waste products. It can temporarily put out transitional molecules during its process of work. Remember, hepatocytes do 4 or 5 different jobs. Now, current theory on that is they don't all do all those multiple jobs. It's they all specialize, but they don't look any different, right? So, depending on what job they do, the space of Dissay could be more functional or less functional, depending, right? So, anyway, that's my best answer for that question. Let's see. Does anybody see what they would call a Cooper cell here? No? Maybe? I'm gonna have to go back and find a couple more slides on this. We have 2 other slide sets we can look at this, and they're probably both better than this one. Give me a second. We'll do this a nice slow way. We got tons of time. See if it's right there by the other note. This is a liver slide as well, and you're going to notice real quick what it looks like. It looks very different, right? So this is not a pig one, this is a human one. So obviously the lobules are much harder to see, and we'll talk about why that's the case in a minute. But what else do you see here that's weird?
Adipose.
You have these, looks like fields of adipose in there, right? That's abnormal, very abnormal in the liver. So what do you think that is? This
Cirrhosis.
is exactly what it is, cirrhosis, right? So this is pathology, right? Histopath, right? This is a standard look of this. So what do you think the ability of that liver to do its main job is done now?
Reduced.
Massively decreased, right? So the more of this damage you have, the less functional that structure is. And remember those like 5 different jobs it does. That's quite a bit, right? But even on this one, when you see let me zoom in on the part that's actually functional and look at the plates and cords, and you'll see that they look much better. Even in this poor specimen here, you'll see that the plates and cords, when you see them, look pretty decent. There's some pathology in there, that's for sure. But you can see the plates and cords a little in there. All right, so we still haven't seen a normal one yet. And this slide doesn't have a normal one yet. All right, so let's look at, what's next? What's on the next page? Triad. So let's look at back to the pig liver for the triad, and we'll see how it looks. So let's grab a triad section and see what we can see.
Sorry, so going back to the human liver, is that those areas of white bits, or is that scar tissue or adipose tissue?
Well, it gets filled with adipose tissue. Remember, one of the things that happens in the liver is what?
Metabolic.
That has to do with lipids, I'm sorry. What process in the liver has to do with lipids? It's not named after lipids. It's named after what? You want me to say it for you? Gluconeogenesis. Converting lipids to glucose. So if you can't convert the lipids to the glucose, what happens? They build up, right? So you start storing them. You make a place to store them. All right, where was I looking at? Triads, okay. So I love this stain. So we're looking at a triad. You want me to get closer or further away? That's not bad.
Not like theirs.
All right, how many things are in a triad?
3.
4. Because that's why it's a triad, right? Why is it a triad again?
It's 3 lobules.
3 lobules come together, right? 1, 2, 3, right? Triad. Not because of what's in it. All right, so there's 4 things in here. What are they? What's the first one? Bile duct. Bile duct's your easiest one. They're by far the easiest one. This is your bile duct right here. It's got a little ring of cells around it, a little hole in the middle. Looks like a little weird donut. Easy to see. What? No.
In my opinion.
Well, there's one.
The arrow on the right-hand side, the one that bifurcates into 2, right? And if you look closely at those, you can see those rings of cells around them. The ones on the bottom from the left there do not have the rings of cells around them. Look very closely at them. I'm going to zoom in a little closer so you can see it here a little better.
Could be a bad grade.
See, now we're too close and it doesn't look as good. Let's leave it this far out. I'll get a regular liver slide in a minute. I'm gonna have to dig one out and find one. But it's got that distinctive ring of cells around it. So we're gonna look at it in the pig and then we'll find a regular slide of it and we'll look at it there. But that's your bile duct, right? What's the second one? Lymphatic. Okay, so the lymphatic and the venule are gonna be very, very similar. The key difference is going to be what, right? For example, this right here and this here, right?
Size?
Not necessarily size, because they can both be fairly large actually, but it's what they're lined with, right? The key is what they're lined with. A lymphatic's not going to really have any lining whatsoever. No real layers or connective tissue, anything, just kind of an empty spot. Whereas a venule is going to have those 3 classic layers that you think about in a vein, right? So on your picture, they're kind of over on the lymphatics are on the left, right? And then that big one in the middle is your venule, right? So this is your venule here, this is your lymphatic down here. There's also, and there can be more than one of each one of these there, right? You can have multiples of each one. So don't think there just has to be one. And then the last one is your artery. Artery on this slide, I don't see a good looking one. This is probably one here, but I don't want to say that without looking closer. I don't see a really good one in that spot. And normally you can see them, but not necessarily. And as I said, I don't really like this stain on this one. There's a good one. I don't know, let me find the other sides. Let's find a regular liver side. See if I have one anywhere. Not in there. Ooh, look, there's a thyroid, parathyroid. We're gonna look at those today, right? Thyroid and parathyroid. Is that on our list? No. You disagree? Things you look for. Maybe I should have you back here. Somebody better out than I. Pituitary gland. We're looking at pituitary now?
Yeah.
We're looking at spleen. Looking at the gallbladder? You would think so, since we're looking at the liver.
We're looking at the liver, hypophysis. No. Nothing just in
Getting close.
Actually, these are both pretty small.
Alright. A few slides, anyway. Let's take a look at this. I've got probably, I don't know. Let me discuss weirdness here for a little bit. I've probably got 7, 8 different slides of different types of things for liver. So for example, this one says liver mitochondria section. So it's stained so you can see the mitochondria in them. Way back in the first lab, remember that? But it's a liver, so it's got the same stuff in it, right? Remember, everything we look at is by structure. So this has a special stain so you can see the mitochondria. That doesn't preclude us from seeing the other things, right? How's this one look? Looks pretty, right? Everybody can see exactly what they're looking at, right? Uh-huh. No? So you don't like this one? Is that what you're saying? Can you explain this one? Of all the things I have. Okay. We'll come back to that. Well, I will find it before the end of the day. I'll go hunt through the whole smash if I have to find what I'm looking for. We'll come back to the triad. What's next?
Hypophysis.
Pituitary gland. Yay. I just had that slide. Trey. All right, pituitary gland. Maybe one of the hardest ones to see for what we're looking at. Remember, it's got 4 parts to it primarily. And you may or may not be able to see them. There's a couple of different stains here. Very different stains, by the way. So this is my lowest power. Not going to be very easy to see for you. So this one, we're up in the stalk right now. This is brain up here, this is stalk here, and then it goes down into the main body of it where you have the 2 halves of it, right? That's, as I said, it's a fairly large organ, right? So that's one version of it. Let me show you a different version of it, then we'll kind of flip-flop back and forth between them. So this one is much lighter in color, but it's also a little bit smaller. So there's your stalk, right? Your infundibulum going down to the main body of it with the two halves, right? So let's look at this one for a little bit just to give us some idea what we're looking at. So the two parts of it are what? What are the two halves of it, shall we say? If you, so let's start first with, there's two different naming methods, right? Actually, there's three different naming methods, but there's two main naming methods. In the lab manual, I have them listed in the pars version. Pars distalis, pars intermedia, pars nervosa, right? You can also call it the anterior pituitary, the posterior pituitary, right? So there's some different variations on what you can call it. So don't get confused by the multiple names for these things, right? Remember that the pars distalis, or the anterior pituitary, is the part that's derived from the floor of the epithelial derivative, right? The pars nervosa, of course, is the one that's derived from the brain, or the posterior pituitary gland, right? Pars intermedia is a part of which one? The pars intermedia is ectoderm derivative, right? And then the stalk, there's a pars tuberalis up there, and then there's also part of the pars nervosa up there. So there's two parts of that as well. So, looking at this one, what do you think that's derived from just by looking at it? Does that look like it's derived from epithelium or nervous tissue? Nervous tissue. That's the pars nervosa, right? That's the posterior pituitary gland, right? On this picture, it's fairly close to what you're looking at. So you can see on the right-hand side of this picture, it looks very much the same. So this right section here is pars nervosa here. So if we slide it over, right, there's your anterior pituitary gland, right? That's the pars distalis. Very different look to them, right? It's easy to spot the difference once you know what you're looking at. And then of course, pars intermedia is going to be between them, like right there, you can see a little bit of it. And this is another one of those things. The pars intermedia is a fairly small structure. And remember, we're sectioning, right? So if you don't section right through the part that has that, you may not have a section of pars intermedia in your slice. So don't be shocked if you don't see one, right? Does that make sense? So where's all the fun stuff, right?
The anterior.
The anterior pituitary has much more stuff than the posterior pituitary, right? Now, they both secrete hormones, but the anterior has much more production as well as much more variation in their structure. So let's look at this a little closer. Let's look at this anterior pituitary a little closer. If you look here, if you look at this section here, what do you see? So the edge is over there. So this is the edge down here, kind of goes like this, right? And this is right in the middle. What does that look like to you? What's the standard arrangement of any organ, really? You got a medulla. And what's this part called? The medulla at the middle. What's this part?
Cortex.
Cortex, right? Are they all going to be that nice? No, probably not. But interesting that the epithelial derivative has that standard arrangement to it. Okay. Remember what the medulla is. It's usually structural, not as functional. All right, so let's look closer at these cells. As I zoom in on them, how many different types of cells are in here?
4.
3.
3, very good.
Okay.
You got the chromophobes and the chromophils, right? And there's 2 types of chromophils, right? Does that sound familiar? Chromophobes, chromophils, and 2 types of chromophils, right? So you've got the red ones, the purple ones, and those pale ones, right? That doesn't look very clear up there. It looks clear on my microscope. I can go up one more without going to oil, so let's go up to the highest power we can.
You said sinusoids are just like tiny little spaces of nothing?
Sinusoids in the liver?
No. Here, like in this picture, I see like 2.
That's about the best focus I can get here. So let's. This is in the pars distalis. This is the anterior pituitary gland. So what are you seeing? What can you see? Just tell me what you got. Well, how many different colors of cells can you see? Remember that the nucleus always stains dark purple, no matter what, right? So what you're looking for is variation in what? In the cytoplasm, right? That's always, when they say that a cell is acidophilic, they're not talking about the nucleus. The nucleus is always going to be purple. So they're talking about the cytoplasm on any cell when they talk about these terms. So for example, how about this cell right here? It's acidophilic, right? It's red, right? The nucleus is purple, but it's got that red corona around it, that red ring around it, where the cytoplasm is. So what do you figure that cell is? Probably an acidophil, right? Shockingly enough. So, what about this one, right here?
Is that a chromophobe?
Or this one right here? You see a nucleus, what do you see for the cytoplasm? Not a whole lot, right? What do you think that is? Chromophobe, right? So what's that leave us with? Basophil. Anybody see a basophil? Well, this one right here. They're not massively dark, but they are dark. So the hard part will be distinguishing between a chromophobe and a basophil, right? Just think, if it's darker, for example, like this one, you can't really see any stain at all in the cytoplasm, right? This one you can see a little, so is that a basophil? Maybe, right? If you had to guess on a test, if I asked you that question on a test, I'd guess basophil, right? Just because, right? This one's better. That's a chromophobe there. That's a good chromophobe. There's a... That looks like a basophil there. Anyway. Some stains in the pituitary gland are better than others. So let's go to that first slide I had and look at the anterior pituitary there and see what you think of that one. This will mess up your day though. So this is the anterior pituitary on this stain. Now this is not an H&E stain, people. You should know that right off the bat, right? Obviously that looks much different than the other one, right? Nowhere near as easy to see. It all looks red like a mess, right?
What kind of stain is it?
So, what are you seeing? Can you define what we're looking at? This one, what can you see really easily though?
The basophils.
Sinusoids, right? You can see those quite easily here, right? Big monster sinusoids, right? Can't miss them. What about, how about this cell? I bet this is a tough one to do, right? Bright red.
The pseudophils.
Probably acidophil, right? What about this guy here?
Basophil.
Chromophobe.
Is that a chromophobe or a basophil?
Chromophobe.
Good question, right? No? Anybody with me? Okay, what about, here, let's do this. How about this one? And this one?
Second one's a basophil.
This one's a basophil, right? Can't tell what you're looking at on which one. This one?
Yeah.
Yeah, this one's tough.
Or I've known that on the paper, I don't really ever.
On the paper.
Okay, the paper's right.
Well, this one?
No.
This one's easy, right?
Yeah.
Why are you wearing that?
It was cheap.
It was cheap. It's not what I said when I went there.
Well, the hoodie was cheap.
He's wearing a Michigan State sweatshirt. That's my alma mater. I think he's just sucking up.
What-
What was I talking about before I got distracted by Michigan State?
Basophils being more of this. So the difference between a chromophobe and a basophil.
She was confused. What are you confused by? This?
Pars distalis.
This picture?
This is just on the paper, I can't tell what's pointing at.
Well, this is the pars intermedia, which is not anything we're looking at right now, right?
Oh, wait.
Does that make sense? We're looking at the pars distalis right now. Okay. I will probably not use this one on the test, if that helps a little at all. I like the other one much better, right?
What's the difference in stain?
This is what's called a BLS stain. It's an interesting stain. I probably should have given that as one of the options for the thing, but it's pretty much only used for the pituitary gland, so it's very specific stain. All right. Let's see. How's it? Let me look at something. It's got an interesting pars intermedia. Okay, so this one, how's the focus out? So that's in focus. So that's anterior pituitary, right? This is the posterior pituitary, or the pars nervosa, right? And then this weird red stuff here in the middle is your pars intermedia. So that's the pars intermedia on this slide. Doesn't look like a picture at all. But it's got the same stuff in it. Let's go to the other slide and see if we can see a pars intermedia. I don't know if I can see one on this one or not. I haven't really looked. Let me see. No, I don't see a good one. A little bit, just a tiny bit. There's just a tiny bit of pars intermedia, and it's right here. Not easy to see either. Can you see it now? See the difference in consistency there? This looks different than this, right? This is just connective tissue, right? This is pars intermedia. This is pars nervosa, pars distalis, pars intermedia. So what's in the pars intermedia? Two things, right? There's cells called basophil cells. And then there's these cysts, right? See the little weird dots? What are those little weird dots?
Basophils.
Nuclei. What is this weird little gap here? I'll bet you can't guess. What's a cyst? Either a gap or a solid structure. And commonly, a lot of things don't stain, remember, so if it looks like a gap, it's either lipids or it's something that doesn't stain, right? Which a lipid doesn't commonly stain. Do I have another slide of it somewhere? See if I can find a different section that looks better. Let's see. How about this one? Ooh, this one looks interesting. Let's look at this one. Here's a different side of it. Somebody used oil on this thing. All right. How's this one look? Oh, that's nice. I like that. Well, let's look at this. This is a different slide, different, same stain as the one slide, different cut by far. How's this one look for staining colors and stuff? First off, where are we? Where is that?
The
Distalis.
Distalis, anterior pituitary, right? Can you tell the difference in this one? No, yes, maybe?
Yes.
No idea, it all looks the same. How about at this magnification?
Good. See basophils.
All right, let's do this then. How about this? This one shouted at me, I don't know why. What's that?
Basophil.
Very good, that's an acidophil.
No. There is. Oh. Right.
Watch what happens to it when I go in and out. See how it's got that weird kind of halo look to it?
Yeah.
That's characteristic of eosin, by the way. Not that you know that. If you slightly unfocus it, you'll usually get a halo glow to it, which is just something that I've picked up over 30 years of doing this, right? So if I do that, right, what do you see? You see this one, you see this one, you see this one, you see that one, you see this one, right? They start popping out at you real close. What about this one here? Pretty solid basophil, right? So, what about this one? Pretty solid chromophobe there, right? All right, let's go to the intermediate one. Let me go down power a little bit.
What were those cells that look close to astrocytes in there?
Close to what?
They look almost, or they remind me of astrocytes. They, like the long, stringy.
I haven't moved it, so let's just zoom
Yeah,
back in.
right there, right in the center.
Right in the center. This
Yes.
blood vessel?
Blood vessel. Gotcha. Why? Okay. Follow-up question. Why can't you see the blood cells within the blood vessel itself? It looks almost solid.
What do you call those in there? I can see the blood cells in there.
Oh, are they capillaries? Are they?
What do you call those? I'm not sure I understand what you mean. Blood cell, blood cell, blood cell, blood cell, blood cell, blood cell. Lots of blood cells.
It's just their stack of
Gotcha. Okay, never mind.
How about this one down here? There's a lot of blood cells. Tons of them, right? RBCs, right? That's a pretty basophil there. Everybody see that one? Shouting at you. This one. That's a nice basophil there. All right, let's see. Pars intermediate. That's what I was going to do, right? On this one. All right. So here you got a nice line, right? Nice distinct line between the nervosa and the distalis, right? And so as we go up here, do you see anywhere as I'm going up here where you have something in the middle.
It's like this.
Like maybe right here. Pars distalis, pars nervosa, pars intermediate, right? What about up here? What's that? You see how it kind of swooped off to the side there? So this is pars distalis, or sorry, pars nervosa, pars distalis, and then this is the infundibulum or the stalk, right? This here is your pars tuberalis. You got a fairly nice picture of it on the next page, I think. This is a dissecting microscope slide image, right? And it shows you your infundibulum and your different parts. Can you tell the parts on this one if you look at it? I mean, they're not labeled or anything, but obviously your stalk's up here, right? And then your parts are here and here, right? What's this part? The main part of this bottom part. Pars distalis. This little weird part on the edge is your tubera or your pars nervosa, right? All right, let's go on to the neurohypophysis or the posterior pituitary gland. Look at that. So let's look over here. This is very uniform in structure and shape. It's not very exciting to look at, so it's very simplistic to look at comparatively. So here it is. It's not very exciting. This is your pars nervosa. This is what it looks like. It looks like nervous tissue, right? It's got that characteristic nervous tissue look to it. And there's only, what, two things in it, right? You've got the unmyelinated fibers, which are the stringy looking things, right? Are you with me so far, right? And then you've got the pituocytes in there, right? Which are the cellular looking things. And everybody can tell the difference, I hope, right? You may zoom in a little closer so you can see them close up. I only have one more I can go, but let's put it up to the max and see what we look like. So here we are, capped out as much as we can be. It's very easy, right? What's this here? Pituocyte, right? And then what are these long, wispy looking things, right?
Fibers.
Yep. What's this again? Blood vessel. Capillary, it's too big to be a capillary. Blood vessel. All right. Anything? What am I missing? Anything? I covered all, I think. Got a lot to cover, unfortunately. We got, what, 7, 8 organs we're doing today. Non-stop fun. Is that what you're saying about this? One lap, right? After the lab practice I gave back yesterday, I'm going right at a big lab today. We don't have a lot of time for this set, right? We have this week, next week, the week after, and then you have a 2-week break and then the lab practice. Or, well, yeah, from the time our last lab to the lab practice is going to be 2 weeks exactly, right? Today's what, 6th, 7th, something like that? So 14th, 21st, and then Thanksgiving's 28th, right? We could have a lab on the 28th if you really want to come in on Thanksgiving Day. How many people are going to be out of state? I mean, that's the day you always go to grandma's house, right? They're not listening. Can I help?
We're trying to figure out, so we're, the Thursday after our Thanksgiving break, is that when our lab practical is?
So, the rest of the semester, right, is what we're talking about, right? So today's the 7th, right? Next week's the 14th. The week after that is the 21st. The week after that is Thanksgiving break. The week after that is dead week, which is when you'll have your lab practical. And the week after that is finals.
When we have our presentation.
No, your presentation is during dead week as well. So you're gonna have your last lab practical and your presentation the same week.
That's what
You picked it, I didn't. But the other option would have been a lab practical on Thursday and a final exam on Friday. Either way is interesting, right? So does that help with logistics? Okay. So now what are we talking about?
Adrenal.
Adrenal gland. Adrenal, right? What's that mean, adrenal?
Adjacent to the renal. To the kidney.
What's it connected to?
Kidney.
Add means added on, right? No? You disagree with that statement? I know I have one here somewhere. I saw it. Thyroid, Golgi, beginning corpuscle. Myocardium. I thought I had a slide with it. Did I not have one? Pituitary. Do I not have an adrenal gland? All right, somebody find me an adrenal gland slide. Here's one, got one. I found it one. Adrenal gland is a cake one, right? Would you agree with that statement or disagree with that statement?
I agree.
So here's the adrenal gland. That's my lowest power, by the way. Compared to the pituitary gland, it's easy. So where are your different parts? This is kind of a thing like the brain, right? In that it's folded on itself, right? So this one, you've got your capsule here, a capsule up there, comes down to the medulla. So this is cortex, that's cortex up there. Cortex on both sides, medulla in the middle, right? If I slide out to the edge where you can see where it folds, see that over there? So medulla down here, cortex, capsule, right? Does that make sense? So what are we gonna look at? You wanna look at the medulla first, you wanna look at the cortex first?
Medulla.
The middle? You wanna look at the middle? So we're gonna look at the middle. Here's some middle. How about this? This is middle. This is medulla. There you go, medulla. It's cake, right? What do you got? It says chromaffin cells and ganglia. What's a ganglia look like?
Round.
Fibers, right? Strings. See any strings? Lots of strings, right? What's chromaffin? Chrome means color, affin means like affinity, like it likes color, right? So color liking is what it literally means. So where would a chromaffin cell be?
Yeah.
Big bright cells, can't miss them, right?
On the handout, it's pretty.
Small, it's a low, its magnification should have been higher. Piece of cake, right?
So, would you agree that the bottom one on our figure there is probably
in the lab slide that you have, the top one is going to be your chromaffin cell.
Okay.
Those two coming out of the bottom are going to be your ganglia. It's not very high mag, no matter what. But as I said, what's the most functional part of it, right? Not the medulla, right? Why do you two keep talking?
She didn't know we did double today.
She didn't know you were doing what?
We have two.
That we were doing two of them today? I didn't realize until I looked at it, but for me it doesn't matter. I don't care if we do 2 of them or 4 of them. I don't have to prep for it anymore. All right, let's go up and look at the, let's look at it, let's see, how about right here? This is pretty decent here. Zoom in there a little bit. Can you see everything? No? Nah. Go back here. All right. Cortex, 3 layers, right? We went over in lecture, right? 3 layers. These are the zona ones, right? Remember your 3 zona. What are they? No idea. It's only labeled on your handout, right?
Fasciculata.
So, what's this thing here?
Capsule.
Capsule. Medulla's down here, so you got from here up to there, right? Is it easy to see the 3 layers? Can you see 3 distinct layers? Piece of cake, right? You got this one right here, you got this band here, and then you got this band there, right? Easy, right?
Yeah.
The outer one, which is the thinnest layer, right, is your zona glomerulosa, right? And when you see glomerulosa, what are you talking about?
Circles.
Ball shaped, spheres, circles, all that stuff, right? They look circular. So that's this one. We'll zoom in on it in a minute. Second one, zona fasciculata, fascicles, lines, right? Columns. If you look at these, you're going to see they are running little tracks up and down, right? And then the innermost layer is the zona reticulara, reticulated, interconnected, meshwork, that kind of thing. And that's down here, right? Same way it is on your lab manual, right? And then it says something about spongiocytes. Spongiocytes are a term they use for the cells of the zona fasciculata. Could you use them for the others? Maybe, but usually it's cells of the zona fasciculata. So let's take a closer look at these 3 layers. We'll zoom in a little bit. All right, so here we are. There's the capsule, right? Everybody can see the capsule. And what do you see here on this outermost layer, right? This outermost layer in here. What do you see? Rounded looking cells, right? Little clusters of round things, right? And if you move down a little bit into this area, zona fasciculata, what's it look like? Lines of cells, right? And these are your spongiocytes here. The primary cells of this layer are your spongiocytes. And then when you get down to the middle, it stops being a line and it starts being just kind of masses of cells. There's still a little bit of the columnar looking to it, but you can see that it's much denser packed and not quite as regular. And that's your reticularis, right? Not hard. Adrenal gland's fairly easy to see the different parts to it. At least I think so. Questions?
Is there going to be 2 separate upload things for each handout, or is there just going to be 1?
Probably need to put them separate. I'll put them separate.
Okay.
All right, let's zip on to the next one. We'll talk about the thyroid, right? Thyroid's easy. In my opinion, thyroid is probably the easiest slide you can look at. I know I have one here somewhere. I saw it. Did you take my tray? That's the spleen. Are we looking at spleen today? No. Spleen must be next time. Spleen not today? Pancreas. Nope, not today. Okay. Well, I thought I had one. Oh, there it is, right there. Staring me in the face, it's okay. Thyroid. Thyroid is unique looking. Nothing else really looks like it, so it should be something you can recognize very quickly what it is, right? I've mentioned that to some of you, is that the hardest part of this is recognizing what tissue it is when you see it. This one is unique, so it's very easy to figure out that you're in that organ. All right. So each one of these ring things is called a follicle. There's tons of them, right? Anybody want to make a guess at what the pink stuff inside the ring is called? Colloid. So we're going to zoom in and take a look at the cells that ring it here. Look at this big one. I'm going to zoom in even more on those cells so you can see them. But look at those. These are what kind of epithelium is that? We use this first lab to look at this epithelium. What is this epithelium right here? Cuboidal epithelium. The key characteristic about cuboidal epithelium is what? Secretory in nature. They're always secretory. Cuboidal cells always secrete something. So the follicular cells are the cuboidal epithelium. I haven't shown you the capsule yet. I will before we get off. Don't let me.
We have 5 markers, but I only see 4 lines.
Say that louder.
We have 5 terms to label, but I only see 4 lines.
Because there's no capsule in that picture.
Ah,
So colloid, follicular cells, right? The follicle, you know what a follicle is, right? That's the whole circle, right? So now let's find something called a parafollicular cell. What does that look like and where is it? What does para mean? Beside. So it's a cell that's not associated with the follicle. So we need to find a cell somewhere. And let me look right in here and show you that, and you can tell me, right? Anywhere in there that you can see a cell that's not associated with follicle. So here's a follicle here, here's a follicle here, right? Here's a follicle here, here's a follicle here. Where's a cell that's not associated with a follicle?
Right in the middle.
That's a capillary, by the way.
Is that one in the top middle one? Very top, like middle.
Anybody see anything that's screaming out different than the other? What one of these? Do your Sesame Street, right? One of these things is not like the other, right?
The thing at the very top in the middle.
This red thing
Nope,
that
right
should
up,
be
no,
screaming at everybody, right? Look how red that is.
Oh.
No, you disagree with that? You don't like the red thing?
Here are follicles.
No. Do I have a good picture of it? Not really. Nothing great. Let's look somewhere else. What if I got a different slide? Let's see. Let me explain something to you here. So if you're looking at this slide, right? This section right in here. See that big band of cells there? Why aren't all these things in here parafollicular cells? Think how this is cutting, right? These tubes are not straight, right? They're not going to go straight line, right? They're all over the place. They're curving all over the place, right? So when you cut them, sometimes you're just going to kind of get the edge of it, right? So what you're seeing here is these are all follicular cells across here. This is actually colloid and lumen here. So you got this weird section of a, so even though it's not, it doesn't look like it's in a follicle, those are follicular cells because you've got an odd cut of it. So you want to look for distinct structural differences and staining differences. Does that help a little bit, I hope?
So is there a parafollicular cell on this, in this, what we're seeing here?
In that whole little area, I don't see a follicular or a parafollicular cell. Well, wait a minute, maybe. No, no. Maybe. Yeah, I see one. You may not think it's the same one I'm seeing, but I see one. Right here. Hey, wait a minute, that didn't look right. That's the size I can go to. See, this is a big mess here. In theory, you should be able to find one here, right? There's a nice one, I like that one. There's a couple of them. Everybody see those? They yell at you a little bit.
Yeah, I see.
Maybe. Everybody see the ones I'm talking about? Yes? You think? These two right here? All right. Thyroid, a piece of cake, right? Not much to know in there. That was my time. We're only an hour in, we got plenty of time. Parathyroid, tougher. I don't know if I have it there. Ooh, that looks like an idea. Hold on. How about that? How'd I know that? What'd I say about parathyroid? No, I didn't say. So, this slide is great. I love this slide because it has 7 different structures in it. And so it's like asking to be confusing, right? So this is a true test of your abilities here by looking at this thing and saying, what is what, right? So what I say about this thing, I just talked about this, right? This, everybody should be able to tell me what that is. Thyroid, that's a thyroid, right? Unique looking, right? So there's a thyroid. Where would you think a parathyroid would be? What did I say it was? What did I say in lecture where it was? Where's your thyroid sit in your body? Right here, right? And there are little things embedded in the back sides of it, right? So where's something embedded in the back of the thyroid in this picture?
Is that small?
Parathyroid, right? Not as hard as you thought, right? All right, so let's talk about some of the other stuff in this picture. What about this thing? No idea. What is that thing? Some weird purple thing, right?
Is that the spleen?
Who said this? Who asked that this test was going to be cumulative? Everything's cumulative in this class, right?
Is that spleen?
So what is it? What type of tissue is that? It's a gimme question. It should be very easy for you.
Spleen.
Because of what?
The
What did you say?
nodules.
She says it's a lymph what? Lymph node, lymph gland. Difference is whether it has a capsule, right? That's the difference between them. Why is it lymphatic? Look at these little ball-shaped structures, right? Characteristic of lymph nodes, right? Plus that dark purple, you can't mistake it, right? Lymphatic, that's a lymph node. Very good. All right, then we get into this. What's this? This red stuff here. What is that?
Pharynx.
Nobody, what'd you say?
Pharynx.
Pharynx? No, it's not pharynx. Should I zoom in on it a little bit? That help? No?
Smooth muscle is blurry for us.
How about now?
Yeah, it's blurry.
You're making it too hard.
Blood, right? I don't know.
What always stains red? Muscle. What kind?
Cross section. Smooth.
That's smooth muscle.
Okay.
All right. We're not done yet. What's this? This is easy, right? What's that? Blood vessel, right? What type?
Large
It's a vein.
Vein. Good idea. Vein. What's this weird stuff here? Adipose, right? Told you, easy, right? What about this?
Skeletal muscle.
That's skeletal muscle, right? See the difference, right? What cut?
Cross.
Cross cut, just like smooth muscle is cross cut, right? All right. Blood, more skeletal muscle. That might be all that's on here. Any nerves, any do I see any nerves? Anybody see any nerves?
No.
I don't, but I'm bad at identifying them.
I didn't really look for nerves, but I didn't really see any either. More smooth muscle. There's the capsule. Hey, look, it's a capsule around a lymphatic structure. What is it?
No.
What's the difference?
Capsule or not.
Capsules around the node, right? All right. There's an artery. Oh, look. What's that? Look, a nerve bundle. Okay. Piece of cake. Where was I? Parathyroid. Parathyroid. Okay, parathyroid. What's in a parathyroid? Two things, right? Two things in a parathyroid. What are they?
chief cells.
Chief or principal cells and oxyphil cells. So, this picture that you have in your lab manual is a different stain than this one, obviously, right?
Yeah.
Obviously, right? So, let me explain some more terminology to you, right? When it says chief or principal, what they're talking about is it's the majority, right? Whenever they say that, it's the majority. Look at this picture in your lab manual. Which one has more cells?
One on the right.
The purple ones, the dark ones, right? So those must be your chief and principal cells, right? Which one has fewer? The big red ones. Those are your oxyphil cells.
Why are they oxygen loving cells?
What's that?
Why are they oxygen loving cells? Why are they called oxyphils? What's their function?
We talked about those in just this last lecture set, didn't we? Did we talk about parathyroid in this set? I think we did. Talked about what hormones it secretes, right?
No one was talking about the pH.
I know we did. I know we talked about parathyroid because we talked about where it was embedded and stuff, right? It'll tell you what those cells do in the lecture if you look. All right, so let's look at this one since this one looks lovely, right? I'm sure everybody can tell me exactly what's what here, right? Should I go higher, Mag? That's about as high as I can go. How's it look?
Alright.
Do you see anything that looks weird and different than the rest of them, or they all look the same?
There's a really long one, like in the middle.
The vast majority of these cells look the same, correct? So what are they?
Chief.
Chief cells, principal cells, primarily. That's the vast majority of what you're seeing there, right? As far as oxyphil cells, I might see one or two little clusters of them, but not much. There's a couple of little clusters that I see in this one, not much of them though. Everybody see them? No? Any idea? For me, it's gonna be lower left and left, right? Should be the same for you, right? It's out of focus a little. All right, chief cells are all these things, right? What looks different? Where do you see something that looks different? On this one. If you look like in here, see how the cytoplasm on these cells looks different? And over here as well. This is not a very good stain. I'll try to find that one. Now, what did I say about the parathyroid?
It's on the back.
It's on the back of the thyroid, but what did I say about it in other organisms? Oh well. Do I have another slide of the parathyroid? I might. Hold on. Let me see what I got. That's pituitary one. Ooh, that's an interesting one. I should show you that one. That wouldn't mess with you at all. Why is that in there? The weirdest things in this slide. So. All right, what am I up to? Is there one here? Ooh, here we go. I knew I had one. There you go. Let's show you this one. This will be fun. So we're still looking at parathyroid, right? Let's look at this one. So what I say about other organisms in the parathyroid? Maybe I didn't. All right, so this is a parathyroid of a frog. How's it look? Same basic structure, right?
Seems like it has more.
How's the color? What's this cell look like to you?
That looks like oxyphil cell.
So it's very easy to see the oxyphil cells here, right? You want to see that on the test, don't you? First, you have to figure out it's a parathyroid, though, right? Which makes it harder. If it's not embedded in the organ like the other one is, because this one's separated out on its own, right? Remember I said that? Okay. All right. I mean, if you look at that lopar, look at it. Parathyroid's all by itself, right?
I still feel like it's pretty distinctive compared to.
All right. Any questions on that? Ooh, let's go to play with the pancreas. That'll be fun. The islets of Langerhans, right? I've got a special stain here. This one's a special stain for islets of Langerhans. Remember I said how many are in there? Lots of them, right? Look at that stain.
That's cool.
Is that wild?
I love that bit.
Look in this section right in here and tell me what you can see. And what do you see? Where's your islets? How many of them do you see? Just off the cuff, I can see about a dozen, right? How about this one right here? And that one right there? And this one right here? That one right there? And that one right there? And that one right there? And that one up there, right? Lots of them, right? Piece of cake. First, you gotta figure out you're in the pancreas, right? Islets usually give it away, though. What's the other thing that's in a pancreas that should give it away that it's a pancreas? No? Let me see if I can find one. I don't see a good one here. That's weird. Usually they're sticking out like you sore thumb. Let me get a different one. This one is not stained specifically for the islets, so let's look at this one. Right? Same structure, very different color, right? But, for example, where's it? Oh, that is a horrible looking slide. Okay. Let me get a different one. This one's better, I can tell just by looking at it. All right, this one's much better. Here we are. Pancreas. What's that big thing right in the middle?
Kidney corpuscle.
The kidney and corpuscle. That's the other key thing that you're gonna see, right? That'll scream pancreas, right? It's only in two places, right? What are the two places where the kidney and corpuscles are? Pancreas and the skin. Well, that's obviously not skin, right? What do you mean no? If you don't know, go back and study the skin a little bit more. That's obviously not skin, right? Pancreas. See islets here? Yes, you can see islets here, right? Everybody agree you can see islets here? How about right, let's see. How about right here? There's one right there. Right, there's one right there. I don't have my glasses on. There's one there. One over there, one over there. And if I put my glass on, I could see more, right? All right, so which one do you want to look at? What do we got for the pancreas? Got two parts to it, right? This time we're looking at, what are we looking at? So let's talk about the acinar glands, right? What's acinar mean?
Round.
That was lab 2, wasn't it? Glands, different types of glands.
It's like the round bottom. The round one.
So if you look at this one, for example, I don't care where we look, anywhere. How about right here? So anywhere that you don't have islets, you have glands, right? Because it's a big gland, that's all it is, right? It's all gland. So all this stuff that you're looking at here is acinar glands. And acinar glands are little circles of cells, right? This little ring here has probably 5 or 6 cells in it, right? So that is an acinar gland. That is an acinar gland. That's an acinar. Each one of those little rings, right? And they don't have to be rings. They can be little long things like that, right? Because again, it matters how you cut it, right? So those are your acinar glands. They're easy to spot, right? What's that up in the corner? Nice size islet up in the corner, right? This thing here, right? See a nice islet there? And go up one more mag and we'll take a look at that islet a little bit. Not bad. I've seen better, but it's okay. So here's your islet, right? They all look kind of funky, right? Do they all look the same?
Yeah.
Kind of, unfortunately. Let's go to the special stain one so you can take a look at that, okay? Let's take a look at an islet. Let's see, let's pick a spot. Ooh, how about that one? That's a nice looking islet right there. It sticks out like a sore thumb. As I said, this is a special stain for the islet, right? I want to do that one. Let me go to this other one. I saw one up here somewhere. There's one. How about this one? Let's look at this one. In some ways, this one's better. Okay. How many types of cells in pancreas? Or in islets, I mean. 3, right? And what are they? So alpha, beta, and delta. And what's a delta shape?
Triangle.
You know where I first learned about delta?
The rivers.
What?
Rivers.
Rivers on maps. Deltas, right? Okay. So, 3 types of cells. You can pick them out easy, I'm sure, right?
Yeah.
You don't think so? They're pretty straightforward, right? This is a special stain just for them, right? That's as high as I can go without going to oil. So what do we got? So here's your islet, right? There's obviously more than one type of cell in there, right? So let's do the easiest one first, right? How about this one? Obviously a delta, right?
It's a delta.
Can't miss that one, it's a delta, right?
Why?
How about this one?
Alpha.
Delta, right? Delta, right? The deltas are easy to see, they don't have any stain in them, right? Delta for does not stain, right? All right, let's go to the next one. How about this one here? How about this one? What color is that cytoplasm? Purple, right? You don't think it's purple? That's not red. It's purple. It's blue or purple, right? It's not red. So what's that?
Beta.
Beta, right? So where's your alphas? No idea. We don't have any alphas, right?
There's like 5.
Alphas? Anybody?
So that one right in the center.
There's a really, I mean, there's several in there, but there's one that's really pretty. One that kind of sits out and you can kind of see all the parts of it. It's not overlaid by some other cell sitting over the top of it messing it up, right? Everybody see it?
Yes.
Right there. Nice and easy, right? No? It doesn't have anything over the top of it. It's got a nice red cytoplasm. You can see the nucleus there. Here's one here, by the way. There's another one there, but they're kind of overlaid by that beta cell there. Here's another one here, but it's again overlaid by this cell. You can see them kind of in the background in this area, but they're kind of covered over by other cells, so it's not as clear, right? You go to a different islet, there's lots of them. It doesn't really matter which one we go to. Let's see, how about this? That's a little one. Is there a bigger one in here somewhere? Do I need to go down power to hunt for them? How do the acinar glands look on this? They don't look bad either, do they? Pretty good. I like them. Let's go down in here, see if we can find a nice islet somewhere. Ooh, that's weird. Okay. Do you see this? Do you see what I'm looking at? I don't have you labeling this on this organ, but does everybody see that? Is it in focus? What do these look like to you? They look like a bile duct, right? From the liver. Let's see, a bile duct carries bile down to the gallbladder, which then takes it to the intestine, right? The acinar glands secrete digestive enzymes that go into a duct that is carried to the intestine. I wonder if it looks similar. So that's what those are, right? Collecting ducts in the pancreas.
Will we be using the special stain or the normal stain on?
You can see them in normal stain, they just aren't as, they don't scream at you quite so much. This is a trichrome stain, by the way, just so you know. That blue for the connective tissue really kind of screams at you, and that's what helps a lot with this one, is that blue connective tissue stain there. Anyway, let's see, what was I looking for? Oh, here's a little islet. It's a little one, but it's fine. How about this one? How's that one look? Not horrible, right? Yeah, no, maybe.
I like it.
Where's your delta? There, I can see 1, 2, 3, 4 of them. 5 of them, maybe. Far left, right? Right in the crease there, right? How about your alphas? That's probably the other
Far
one.
right.
There's a little cluster of them over on the far right side. 3 or 4 of them over there. Everything else is betas, right? Not bad. Questions, comments, statements? You want this on the test, you want the other one on the test?
This one, sure.
How about this one? There's a nice islet there.
That's actually a really nice one. You can see them all very clearly.
Mm-hmm.
It's not bad. Not bad at all.
All right, where was I? What was I doing? What's next?
I think that's the main one.
Is that it?
Mhm.
How many organs?
8.
5, 6,
Okay.
7?
That's a lot.
Well.
So, everybody needs to study for this lab practical and get a better grade than you did last time. Everybody.
Can we do like key rentals to this building? Like just so like us in the histology class can get in here like later on. Some of
I'm gonna get my secretary to block me out some time so I can come in here and get some trays of slides out for you for everything that's gonna be on this test set. And I'm gonna take all those other slides and put them in the back, and I'm gonna have a little row of slides over there that you can use to study from.
That would be awesome.
I will work on that, I promise to do that. I will tell my secretary as soon as I go back to my office to block me out some time to do that next week, okay? All right, that's all I got for you. I saw that. We have to talk about it.